Hi, and welcome to the VieSID Press-release. In this video, we report on articles selected from the literature published in August 2023 that we hope might be of interest to the VieSID community. So let's begin. In Archives of Oral Biology, De Caux et al. Published a systematic review addressing the issue of cortical bone distribution of the mandibular corpus and symphysis and the loading environment. They describe the biomechanical scenarios currently accepted, explaining the types of elastic deformation to which the mandible is subjected during chewing. As seen on the right side of the image, unilateral molar biting at the working side results in a positive sagittal bending of the mandible, a negative anteroposterior torsion, which consists of an inversion of the basal border, a positive sagittal shear, and the lateral transverse bending, also referred to as wishboning. Positive sagittal bending leads to tension at the lower border and compression at the upper border of the corpus, and constitutes the predominant deformation occurring in the working side during unilateral biting. The balancing side experiences forces in the opposite directions. Lateral transverse bending or wishboning leads to a compression of the buccal part and tension in the lingual part of the symphysis. Frontal bending produces a compression of the alveolar part and tension in the inferior part, as well as torsion between the loaded and balancing sides, resulting in dorsoventral and anteroposterior shear. In 1984, Demes et al. Suggested that in the postcanine corpus, and especially in the molar area, shear forces resulting from occlusal forces affect the buccal cortical bone, while mixed shear forces and counteracting torsional forces affect the lingual cortical bone. On the lingual side, the addition of shear forces and counteracting torsional forces results in reduced strain on the buccal side. Since this hypothesis is still debated, the authors performed a systematic review of the literature and meta-analysis, considering the articles focusing on the relationship between the distribution of the mandibular cortical bone and mandibular bone function in humans, including papers from 1984 to April 2022. They excluded experimental studies on animals, as the human mandible is peculiar for the unique pattern of masseter recruitment, for a more vertical-oriented symphysis, and for the presence of a chin. After article selection and screening, and after an assessment of the risk of bias, the authors considered a total of 20 articles. This showed various study designs, including mechanical experiments, finite element analysis, cross-sectional studies comparing different subgroups based on their bite forces or based on their dietary habits, and comparative and cross-sectional studies comparing subjects with different facial patterns. Collectively, the selected articles addressed whether mandibular cortical thickness distribution reflected cultural and dietary habits, facial divergence patterns, or bite force, or specific loading regimes. The main results of this systematic review were summarized in their figure 4. Here you can see in A, the mapping of the predicted cortical thickness in the postcanine part of the corpus on the buccal side. Cortical thickness increases from canine to third molar, thus following an anteroposterior gradient. This gradient of thickness is correlated with a similar gradient of strain during function, particularly in the superior part of the corpus. B shows the mapping of the predicted cortical thickness, which presents an inverse or uneven pattern compared to the buccal side. Lingual cortical thickness is thinner than the buccal side, and this buccal lingual gradient is more pronounced for the molar and upper areas of the corpus. This buccolingual cortical thickness gradient mirrors the buccolingual gradient of strains in experimental studies under torsional and shear conditions. C represents the area experiences a positive relation between facial divergence pattern and cortical thickness, while the area represented in D does not vary between faces with different degrees of divergence. The authors concluded with a take-home message for clinicians stating that hypodivergent patients present significantly thicker buccal cortical bone in the molar areas than hyperdivergent patients, and this should be taken into account while applying mini screws or during surgery. For the 50th anniversary from the establishment of community dentistry and oral epidemiology, this journal published a special issue focused on inequalities and inequities in oral health care. As stressed by the guest editors Borel et al., the burden of oral diseases such as dental caries, periodontal diseases, tooth loss, and oral cancer is disproportionately carried by the individuals of minoritized and disadvantaged groups in a society across country, regardless of their level of economic development. The articles collected in this special issue discuss various topics, including the equity or better inequities as a matter of social justice, where communication is considered a tool for knowledge transfer from academic or non-academic institutions within and between countries to address inequities. Systems and forms of oppression, such as racism and its consequences at the societal and individual levels. Oral health inequities across the life course of individuals in several countries. Borel and the other guest editors concluded that systemic inequities around the world are avoidable and preventable, yet persistent. Most of the determinants of poor oral health are structural, and attention beyond the social factors is necessary. Thus, political and commercial determinants of health must be included in the discourse on oral health inequities and inequalities. Lockerman and Hauser wrote the Cranio guest editorial on the association between mandibular position with respect to the cervical spine and the influence on the internal jugular vein diameter. In this article, the authors discussed the diagnostic procedure that Dr. Ross Hauser applies at the Hauser Neck Center in Florida, USA, to test whether patients need to be referred for spleen therapy. In particular, symptoms such as intracranial hypertension, dizziness, vertigo, migraine, nausea, and tinnitus can be caused by a reduction in the diameter of the jugular vein, which represents the main drainage system of the brain. Compression of the jugular vein can be caused by cervical destructure, upper cervical instability, elongated styloid, or in some cases, mandibular displacement. This situation seems to be quite common. In fact, in a 2012 study by Jayaraman et al., 108 consecutive patients underwent CT angiography for presumed arterial obstruction. 50% of them were found with internal jugular vein compression, and for most of them, the compression was at the level of C1. Usually, venous blood drains from the brain through the internal jugular vein, but if that is blocked, as it could be in approximately 6% of the population, blood is drained through the vertebral veins, external jugular veins, and cervical veins. To verify whether the mandibular position is responsible for the decrease in diameter of the internal jugular vein, Hauser recommends high-resolution ultrasound in upright position with different mandibular positions. If the diameter increases in mandibular protrusion or with tongue tip against the palate, the patient is recommended for mandibular repositioning. Differences between individuals with different skeletal configurations can be appreciated also using CBCT images. Also, the authors speculated that some improvements in the patient's health following mandibular repositioning to enlarge the airway might in fact lead to a positive outcome in case this results in an increase in the diameter of the internal jugular vein. Thus, they recommended that in case of symptoms possibly associated with the constriction of the internal jugular vein, proper diagnostic is performed to evaluate whether mandibular advancement is recommended. The Journal of Dental Research published an experimental paper by Zhang et al. Investigating the physiology of destruction osteogenesis at a cellular level. The authors contraposed two models, that of bone defects caused by trauma and that of destruction osteogenesis. The latter is a highly effective procedure for bone regeneration, although its actual underlying mechanism is still poorly understood. Thus, Zhang et al. Conducting this animal experimental study to investigate the phenomenon and established destruction osteogenesis and bone defect models by osteotomy of canine mandibles. By means of microcomputed tomography, they showed that destruction osteogenesis led to new bone formation. In contrast, the bone defect model demonstrated incomplete bone union. Through histological staining, the bone calluses from the two models were isolated and identified, showing that mesenchymal stem cells from the destruction osteogenesis model possess a stronger osteogenesis capability. They then performed the single-cell RNA sequencing and found out that in the destruction osteogenesis group, two subpopulations of mesenchymal stem cells called PRRX1+ were present that expressed the marker of neural crest cells and were associated with the process of epithelial mesenchymal transition. By means of immunofluorescence, they corroborated the results in vivo and in vitro, experimentally validating that continuous destruction maintained these mesenchymal stem cells in an embryonic-like state. When the ability to produce these mesenchymal stem cells was genetically impaired, jaw bone regeneration was inhibited, and the ability of osteogenesis, cell migration, and proliferation diminished. Taken together, this study provided a novel comprehensive atlas of the cell fates in the context of destruction osteogenesis regeneration. In the Journal of Dentistry, Chen et al. Analyzed the association between tooth loss and cognitive decline in the elderly, testing the mediating role of high-density lipoprotein, or HDL cholesterol. Previous research has highlighted an association between increased levels of cholesterol and cognitive decline. At the same time, recent research has found an association between high levels of cholesterol and tooth loss, and animal research has shown that high levels of blood cholesterol are associated with alveolar bone loss. Conversely, HDL cholesterol seems to have a positive effect on cognitive abilities. Thus, the authors performed a retrospective assessment of data retrieved from the public database of the National Health and Nutrition Examination Survey. They included patients' data from 2011 to 2014 from patients of 60 years old or older. Datasets were further selected based on the presence of tooth number determination, cognitive functional test, and cholesterol level, and for completeness of information. Eventually, a total of 1,124 patients could be included. Tooth loss was classified as per the World Health Organization recommendations. The control group was constituted of individuals which had 20 or more teeth. The moderate tooth loss group of individuals with 1 to 19 teeth, and the severe tooth loss group of individuals with no teeth. Cognitive test considers were Alzheimer's disease immediate recall test, delayed recall test, animal fluency test, digital symbol subdivision test. The authors accounted also for additional variables and found a positive association between tooth loss and age, functional decline, smoking, ethnicity, income, and education level, but did not find an association between tooth loss and cholesterol level. Chen et al. Then explored the effects of tooth loss on cognitive functions. They built 3 different models. We now focus on the third model, in which demographic factors such as smoking, education, income, ethnicity, and marital status were controlled for. In this model, moderate tooth loss was associated with worse delayed recall outcomes. Moderate and severe tooth loss were associated also with inferior motor processing speed and complex attention. They then performed a mediation analysis to test the impact of HDL cholesterol levels on the association between remaining tooth number and cognitive function, and found that serum HDL cholesterol levels were positively related to the immediate recall test and the delay recall test and the motor processing speed and complex attention test. The indirect effect of HDL cholesterol levels in tooth number for immediate recall and motor speed abilities accounted for 5% and 2% respectively. In conclusion, this study seemed to suggest an association between tooth loss and cognitive function that is independent of age and other confounding factors that might be mediated by cholesterol. However, further studies exploring the causal relationship between these factors are of the essence. If the role of blood cholesterol levels in accelerating both tooth loss and cognitive decline will be confirmed, measures from control of cholesterol levels should be put in place to prevent tooth loss. In the Journal of Oral Rehabilitation, Minervini et al. Investigated the effects of economic inequalities on temporomandibular disorders by means of a systemic review and meta-analysis. Socioeconomic factors have been considered one of the etiological determinants for temporomandibular disorders. Thus, the authors screened the literature for papers on the topic, considering human subjects reporting low economic status and compared to medium and high economic status in relation to the prevalence of TMD. From an initial count of 153 articles retrieved, only 3 articles could be considered. This search, therefore, led to limited evidence. One of the articles by Johnson et al. 2006 compared prevalence in blue collar versus white collars on a total of over 12,000 subjects and found only a minor difference in prevalence, 33% versus 12%. The other 2 papers compared low versus high income samples and found a higher difference in percentage, although in the paper by Magalhaes et al., only a sample of 100 subjects was considered. But De Souza et al. Considered over 2,400 individuals and still found a higher prevalence of TMD in the low-income group. Indeed, this article showed that women and those who reported poor general health, dissatisfaction with dental care and their teeth, dental fear, bruxism, overbite or overjet, and those with removable dentures had a significantly higher risk of both TMD pain and dysfunction. Based on this study, I would conclude that although the association between TMD and socioeconomic factors seems highly likely, systematic epidemiological studies should be performed to appreciate the extent of the socioeconomic influence of the development and maintenance of TMD. Orthodontics and craniofacial research published a paper by Torres, Tagawa et al. On the position of TMJ disc in skeletal class 3 patients. This cross-sectional observation study included 78 boys and girls. They were patients of the Department of Orthodontics in the University of Santa Cecilia in Brazil. They possessed angle class 3 or class 3 subdivision malocclusion and or normal occlusion. The position and the shape of the TMJ disc were evaluated based on MRIs. Subjects with normal facial configuration did not present a malposition of the disc. Instead, in class 3, the discs were displaced in 30% of the cases, and in class 3 subdivision in 12%. Anterior and anteromedial displacement were the most common types of displacement. Almost all subjects showed biconcave discs L2 class 3, and the control group showed also a low percentage of non-biconcave discs. Disc position and shape were not dependent on facial proportions. This study showed that class 3 patients are at risk for TMJ disc displacement and should be therefore considered for early intervention. We've come to the end of this presentation. I invite you to contact us in case you want to know more about our activities. Otherwise, be ready for next press release. Best wishes to all.